Welcome, everybody, to this week's edition of the Extracellular Vesicle Club. It's my pleasure to introduce our speakers today, who are Martijn van Herwijnen and Marca Wauben from Utrecht University in the Netherlands. They're going to be talking with us about some work that they've been doing for quite some time. They're really some of the world's experts on EVs that are found in milk. And so we'll be hearing about a recent publication that they had in the Journal of Extracellular Vesicles that explores what milk can do to barrier function and then also to immune responses. So I'd like to hand it over to you, Martijn, and thanks so much to you and Marca for joining us today.
Well, thank you, Kenny. It's an absolute pleasure being here and being allowed to present our paper in the Journal of Extracellular Vesicles. So the nice thing about this setup right now is that I can go through the main figures of our paper, but at the same time, I can also show you all this supporting data that is sometimes a little bit hidden away and not everyone sees it. So I can really use this format now to show you the complete picture of our story. So starting off with this slide, it's a nice opportunity for me to also acknowledge the people that contributed to this paper. So obviously these are the co-authors of our paper. So I'd like to acknowledge some people from the start. So this work was conducted in the group of Marca Wauben. And together with Esther Nolten-het-Hoen, a couple of years ago, we started this milk EV research. And Marijke Zonneveld, who was at the time a PhD student, who's now a postdoc at Maastricht University, worked on this project together with myself. And Marijke and I, we share the first authorship of this paper, and that is why I'm presenting you with this work. We got a lot of funding, obviously, and we need to acknowledge that. So part of this work was funded by the Dutch government in a partnership together with Nutrition Research, which is a daughter company of Danone. We also received funding from our own university via the Future Food Utrecht. And currently we are involved in 2 EU projects called EV Foundry and Train EV. And part of that work is also conducted on milk research. And finally, I need to acknowledge the milk donors who were graceful enough to share us some spare milk that we could use for our studies. So when you think about milk and breastfeeding, the mother is not only providing nutrition to the infant, but there's a lot of communication going on. And milk is a very complex biological fluid in which basically all the milk components have their own function. You can find various macromolecular structures in milk. So for instance, you can find cells in milk. These can be exfoliated epithelial cells or immune cells or even stem cells. Next to that, you have casein micelles and milk fat globules. And these are, in my opinion, really the nutritional components of milk because they provide the amino acids and the lipids that are needed for growth. Then you have what we call the whey fraction that contains soluble molecules. So you can think about antimicrobial peptides or antibodies. So if a mother gets vaccinated against corona, she will have antibodies directed against the virus in her milk. And also you find, for instance, carbohydrates in milk that can be used by the infant's microbiota to grow. Milk itself is not sterile, so it also has its own microbiota. So you can find bacteria, but there are also recent publications that show fungi and even viruses naturally present in milk. And then finally, you have your extracellular vesicles. Now, if you think about nutrition and immunity and where this might act, most people think, so nutrients are taken up in the gut. So probably also all the immune regulatory functions are happening there. But we would argue that you should not oversee the oral cavity because that is the first entry point of the milk. And that is the first site where these milk extracellular vesicles might interact with the infant. And it's also an important immunological site because when you look in the oral cavity, you have your gingival epithelial cells, but underlaying this epithelial barrier, you can find immune cells. And these can be cells from the innate immunity. You can have antigen-presenting cells like dendritic cells, but also adaptive immune cells like T cells. So also there at this anatomical site, a lot is going on. Now, I'm going to talk about vesicles a lot, and this is the EV Journal Club, but yet I anticipate that maybe we also have some milk researchers in the audience that are not really familiar with vesicles. So slightly something about that. So living cells, they produce vesicles, and these are various subsets. So from the plasma membrane, you can have budding off vesicles that we typically call microvesicles, or you can have exosomes that are formed in multivesicular body, and then they are released in bulk. But once they are secreted in the extracellular milieu, we call them extracellular vesicles, because there is basically no distinctive marker or characteristic that can fully discriminate EV subsets. So that's why we tend to just call them by a common name, extracellular vesicles. And what you can see from this picture is that they are heterogeneous in size, but also in composition. And this is just from one cell type. Can you imagine that in milk you have dozens of cell types that could be the potential producers of milk EVs? So there, the heterogeneity is probably even bigger. So these nano-sized vesicles, they contain a bilipid membrane that is often enriched for certain lipids. Within the membrane, they can have transmembrane proteins, for instance, the tetraspanins, CD81, CD9, and CD63. In the lumen, inside the vesicles, you have proteins, but also other material like metabolites, amino acids, or genetic material. And I think microRNAs are one of the most known genetic materials that are being studied in vesicles. So these vesicles, they are sent to other cells and they are taken up, and that can happen via various routes. So it can be receptor mediated. Maybe they just sit on the surface and they start signaling immediately. They can be taken up via various mechanisms, or they can simply fuse with the plasma membrane and thereby releasing their content into the recipient cell. So I'm going to talk about milk EVs, obviously. So in order to study them, we need to separate them from other milk components. So we are now coming to figure 1 of our Jeff paper. And actually, this figure is sort of a summary of a publication that we had a few years ago, also in the Journal of Extracellular Vesicles, because we spent quite some years trying to figure out what the optimal separation procedure would be. And we found that when you start with fresh milk, this really complex biological fluid, you have these cells and these milk fat globules in there that are actually quite unstable. And you want to get them out of the milk as soon as possible. So that is what we do. We typically get the milk in our lab within 20 to 30 minutes after expression. And then we do a centrifugation step at 3000 G, which allows us to pellet the cells. And in the same way, on the same time, you also get a cream layer that contains the milk fat globules. You can separate that, and after that, you get this supernatant that you can store in the freezer for long term. So my advice to people that work on milk and want to study milk EVs is do not store whole milk, because these cells, they fragment. The milk fat globules are very unstable, so you introduce artificial vesicles, and you're no longer studying these native vesicles that are in milk. So then we proceed with the 5,000 and 10,000 G centrifugation step. And what is typically done in EV research is that people make 100,000 G pellet, for instance. But for milk, this is really tricky. And that has to do with these casein micelles. Because if you make 100,000 G pellet, the pellet is really solid. It's really hard to resuspend. So what we decided to do is take this supernatant and put it on top of a gradient. In this case, in this example, we're using OptiPrep. We spin overnight, and what happens is that the vesicles will obviously go into the gradient and they will stay at a certain density. We can take out those fractions, and what we then do is we put this on a Cephadx column, and by using size exclusion chromatography, we exchange the OptiPrep for culture medium. And that makes us EVs ready to use in our cellular system. But this is not yet enough, because you need a control to see if the effects that you are measuring are an EV-mediated effect. So that is why we came up with our control, which we call the EV-depleted control. What we actually did is we took this protein-rich milk supernatant that no longer contained EV and put this on top of a new gradient and repeated the procedure. So in this case, we have a procedural control that if we see any effects here, we know that it's not an EV effect, because basically what we expect is that the EV sample should contain pure EVs, while the EV depleted sample should be devoid of extracellular vesicles. Now, one important thing to note is that when we load sample on top of the gradient, we put 6.5 ml of milk supernatant on top of the gradient. And after isolation, we end up with 7 ml of extracellular vesicles in culture medium. So we describe this as a near physiological concentration of EVs. And we try to add as many EVs as possible to our samples. And what is also important is that we do not measure, for instance, protein content, or we do not correct for particle numbers. We use the milk as it is because it's a natural resource. And the infant determines how much milk it ingests, but when it enters the oral cavity, it is at that concentration in which the cells are exposed to the milk. And that is why we like to use the same concentration of EVs together with our cells. So obviously, in order to do this research, we need to characterize the EVs to make sure that we're really working with pure EVs. So we started off with an NTA to look at the particles because we still wanted to have an idea how many particles we could find in our preparation. Well, it is no surprise that we find a lot of EVs in our EV sample, but very few in our EV depleted. And actually, although these EVs come from human donors and you expect some variancy, they look quite similar also in size. And when you basically look at them under the microscope in the NTA, you can nicely see these particles over here that are probably the milk EVs, and in the EV depleted, you don't see anything. We took great care to check if the NTA was running properly so that we have enough particles in frame, but also enough valid tracks to be really sure that the particle numbers that are over here are actually correct. Now, you might wonder, if you are more familiar with milk, why these numbers are so much alike, because you might know that milk changes in composition over time. But actually, these samples came from mature milk. And that means that on average, the women had given birth 7 months ago. So that means that the milk composition is not likely to change as much anymore. And it could be that if, for instance, we look at early milk, these numbers do change and maybe there are more vesicles in there. But these were the samples that we worked with. We also did a immunoblot assay to see if we could find EV-associated markers. So we find the tetraspanin, CD9 and CD63. We checked additional markers like flatidin-1 and heat shock protein 70. And we also checked what we would describe a negative marker for EVs. So this is lactoferrin, which is an abundant milk protein. And actually what you see is that it is present in the EVs, but also in the EV depleted control. So this procedural control is not completely empty. And I think it's really good to have this control because, again, if we see an effect with our EV sample, we need to make sure that it comes from the EV and not some kind of background. Another thing that I really like about this Western blot is that you can see the differences between the individual donors. For instance, there's a difference in glycosylation, or there's a difference in the amount of marker that you can find between different donors. So it really shows you that the mothers have a unique profile of EVs within their breast milk. So now we move to the functional part of the paper. So first of all, we wanted to look at the barrier function because the barrier is very important, especially during the first months of life. And it is already known that human milk supports this process. So we wanted to know, do milk EVs support this process or do they play a role? So what we did is we took a gingival epithelial cell line that we cultured into a monolayer, and then we made a scratch. And with that scratch assay, you can look at, which is a model for barrier function. So you can add your EVs or your controls, and then you can look within 3 to 5 hours how the gap closure is affected by anything that you add. And this inhibited or enhanced gap closure is a measure for barrier function. So that is what we did. And these are the videos that we got that are in the supplementary files. So it is a cell line, so it's always growing. So in the medium control, you see that there's a certain rate of gap closure. Now we can add a positive control with TGF alpha, which is a growth factor, which basically allows the cells to grow faster and they push each other into the gap. With the EV depleted, gap closure was not really enhanced, but with the milk EVs, we could actually see the gap closing faster. Now, if we quantify this into a figure in which we measure the number of cells that enter the gap per minute, we see that with the medium, there's a certain background. With the TGF alpha, it is the highest, but actually the milk EVs perform quite well. So apparently the gap closure is enhanced. Now, this raises the question, how does this work? What is their mechanism of action? So when we go back to this picture of this EV, you can see that there's a lot of content in these EVs, and there could be a lot of different molecules that are involved in this gap closure. So we thought, how are we going to tackle this? And we thought back about a paper that we published a few years ago in which we characterized the protein content of our human milk EVs. And actually what we found is that when you look at the complete human milk proteome with about 2,600 proteins, a large proportion of these proteins are also found in milk EVs, but we actually identified over 600 new proteins that no one else had yet found in milk that come from milk EVs. So they are an important part of the milk proteome. And with this data set, we could look into what the contribution of proteins would be to the gap closure. Now, this study was performed on 7 individual donors. So all the data we put in here was also from 7 donors, but we wanted to know what the core milk proteome was like. So we only took the proteins that we identified in all 7 donors. And the first thing that we did was, are these proteins in these milk EVs, are they just singular proteins, or do they maybe form clusters? So by using string, we could actually identify that many of the milk EV proteins that we previously identify are likely to form protein clusters. And this is interesting because that would mean that if a recipient cell takes up these milk EVs, it gets a cluster of proteins. And if you can do Go analysis on this, you can actually see that some clusters are more involved maybe in proliferation or in migration. So you can really get a lot of information about this. But there was one disadvantage though about doing the Go analysis, because with the Go analysis, you can look and screen for migration, because that maybe is part why this gap closure is enhanced, or proliferation, because maybe they're pushing each other into the gap. But it doesn't say you anything about what individual proteins are doing. Are they enhancing the migration? Maybe they are inhibiting the proliferation. So then we started a very tedious task because we checked actually all the individual proteins for what they can do. And we came up with this list that we put in the supplementary files. We have our milk EV proteins. And for instance, we could see that, for instance, this protein is bound by CDC42 and REC1. Those are involved in migration, and it targets actin. So that makes sense because if you want to move as a cell, you have to reorganize your actin cytoskeleton. But what we also saw were some proteins that are here shown in yellow that we could not find any evidence that they are involved in those particular signaling pathways, although according to the Go analysis, they were annotated to be involved. So we decided to exclude these proteins because we couldn't work with them because we wanted to know what happens in the signaling cascades. So in the end, we started building these huge networks of potential interactions that you could see. So this basically represents a cell with a cellular membrane. And in blue, you see the milk EV proteins that we previously identified. But now we're looking at where in the cell could they act. And then in white, we have the cellular proteins that are needed to complete the signaling cascade. What we saw is that there were many different potential interaction possibilities of the milk EV proteins. And we also saw that around a few nodes, there were a lot of interaction possibilities. So these hotspots appeared. And for one hotspot, for instance, here, that has to do with proliferation, there was a lot of inhibition. While in another hotspot that has to do with migration, there were also a lot of positive signals. So in the end, we decided to condense this complicated graph into a simpler representation of what we think might be going on. So you have your cell, you either have migration that is involved in the gap closure or the cell cycle that is involved. We have all these milk EV proteins that could potentially interact at different levels in the signaling cascades. But we saw actually that there's the likelihood that the cell cycle is inhibited unless the signaling was going via MEC1 and 2, or that the migration was activated and enhanced. So what we did is we decided to use inhibitors of these pathways to show if these pathways are involved in the enhanced gap closure. So when we inhibited MEC1-2, we saw a slight decrease of the milk EV effect, but it was not significantly different. However, if we inhibited P38, our milk EV effect was completely abolished. So this strongly indicates that P38 and thus migration is involved in the enhanced gap closure. So when we submitted the paper, the reviewers were not yet convinced that this was really going on. So we had quite some discussion about it. I have to say they were very constructive and also very fair. So it really improved our paper because we decided to go back to our videos to track these epithelial cells during the gap closure. And what we were able to do is, we could track individual cells over the 3 to 5 hours that we were doing the experiment, and we could track the amount of displacement that they showed. So we did that for all the conditions. So I'm showing you here the EV, and you compare that to the EV depleted, and there's just less migration going on. And when you quantify this, you can actually see that on average, cells that were cultured in medium didn't have that much displacement as well as the EV depleted condition, while in the TGF alpha, they did have a lot of displacement, but also in the EVs. So this data really supports that migration is more dominant over proliferation. And then finally, we decided to do another experiment because when we went back to our complicated model, we could also see that various adhesion molecules might be involved in these epithelial cells, especially E-cadherin popped up as a hotspot. So because we were doing these experiments in a time window of 3 to 5 hours, we did not expect de novo synthesis of new proteins in such a short time. But what we hypothesized is that these epithelial cells, they have their adhesion molecules upregulated because then they can really connect and form a strong barrier. But if you want to move around, you actually have to downregulate these adhesion molecules because then you can move faster. So that's the experiment that we did. We just exposed these cells to milk EVs and we checked E-cadherin and EpCAM. So what we saw is that almost all the cells express these markers. But when you look at the geometric mean, you see that in the conditions where we expose them to milk EVs, the amount of markers was lower per cell. So this suggests that they downregulate these adhesion molecules, and that might allow them to move faster during the gap closure. So we looked at the epithelial barrier, and then we moved to innate immunity because obviously the GI tract is exposed to microbial, dietary, and also environmental components. For instance, epithelial cells express toll-like receptors that are important for innate immune signaling. So we asked ourselves the question, can these human milk EVs affect the TLR signaling? So we started with a reporter cell line, which are human embryonic kidney cells that expressed either TLR receptor, that we can stimulate them with their agonist in the presence or the absence of milk EVs. Now, just as a small reminder, these TLRs, they all recognize their own ligands, and some of the recognition can be quite broad, or they can even recognize self, like TLR2 and TLR4. Another important thing is that these TLRs can either be expressed on the surface, like TLR2 and 4, but others, like TLR3 and 9, are in endosomes inside the cells. So that also has to do with how they function and what pathogens, for instance, they need to sense. So we do these experiments in this reporter line. So in the end, the reporter line produces alkaline phosphatase. So the more of this phosphatase you find, the more activation there is. So when we look at TLR2 and 4, if we stimulate them with an agonist, we get 100% response. And what we saw actually is when we stimulate them with EVs, and even for TLR4 with the EV depleted, we see some effect. Now, I'm not showing you these data, but if you look in the supplementary files of our paper, you can already identify or see that we show some human milk EV proteins that are self-antigens or self-ligands for these TLRs. So it could happen. But we also got the question from the reviewers, are you really sure that your preps are free of any bacterial products? And as I showed you in the beginning, in the introduction, I say milk is not per se sterile, but we checked for LPS, for instance, in our EV prep. And what we saw is that indeed the EVs, they did contain endotoxins, while the EV depleted did not contain that much. So again, this shows that the samples that we have are not per se sterile, and that they do contribute or can contribute to the responses that we see here. Now, in the second part of the experiment, we also did a stimulation together with the ligand of the TLR and the EV of EV depleted control. But for TLR2 and 4, we didn't see any differences. However, when we did the same experimental setup for TLR3 and 9, we actually saw that in the presence of poly-IC or CpG, the response was inhibited. So there seems to be a very specific modulation of these endosomal TLRs, but not the cell surface TLRs by these milk EVs. So then we moved on with the next experiments. We moved away from the HEC cells and we went back to our gingival epithelial cells. These cells respond to TLR, or they have TLR3, so they respond to poly-IC. We stimulate them and we expose them to the EVs. These are not reporter cells, so we decided to look at the messenger RNA of IL-6 and CXCL8, because these are known cytokines to be secreted by these cells. What you see is when you stimulate them with the poly-IC, there's the upregulation of messenger RNA, and in the presence of EVs, the messenger RNA upregulation is inhibited. But for us, this was not enough. These were only 2 messenger RNAs, so we decided to do a bigger PCR array for TLR-related genes. So that is presented in figure F. And for this experiment, we also included the EV depleted control. And what we actually saw is, for instance, there are conditions where the medium gives hardly any response, while the poly-IC does, and the EVs inhibit this, for instance, the IL-6. We also see some conditions where the EVs suppress a little bit, but not fully, like the CXCL8. But it could also be the other way around. So, for instance, for these 3 genes, we find more messenger RNA upregulation in the presence of EV. And the funny thing is that the SIGR and SARM1 are actually negative regulators of TLR signaling. So the milk EVs turn on negative regulation. And then finally, I think a lot of people tend to forget this, but even in medium conditions, you can get upregulation of certain messenger RNAs, which then are downregulated by other conditions. So this was very interesting for us, and it helped us to further understand what might be going on. So we went back to the proteomics and we came up with this scheme where we said, well, for these 4 TLRs that we checked, in the end, you can look at cytokine production as a downstream effect. But since all 4 of them diverge to this singular pathway, and we see these differences between the membrane-bound and the endosomal TLRs, we expect the regulation not to happen here. So there must be going on something in these endosomes. And at that point, we actually only identified one protein, cystatin B, but it is known that it inhibits the cleavage of these endosomal TLRs. So we checked it with immunoblotting and it was there on the EVs and not on the EV depleted control. But we believe that there might also be something happening in the trafficking of the TLRs to the endosomes. So something is going on there. But what I noticed when I was doing the analysis is that when you look at the literature, a lot is known about this part of the signaling, but not yet a lot is known about how trafficking and cleavage is regulated. So I bet that in a few years, new studies come up and I can probably better understand what's going on here. In the end, we decided to look at the TLR3 to see what's going on there. Is it cleaved more or less, or is even the full length changed more or less? So what we saw is that when we look at the quantification, in the presence of EVs, there's less full-length TLR3, but also less cleaved TLR3. And this cleaved TLR3 is important for the signaling. And we had actually not expected that the full length would also be lower, but apparently that is what the milk EVs do. So there is already a regulation in the amount of protein that is present. And this is different compared to the positive control and the EV depleted. So in the end, we show that these endosomal TLRs are regulated by the milk EVs and not the cell surface TLRs. So that's the part of the innate immune responses. So then we switch to adaptive immunity, because it is known that EVs can interact with a variety of immune cells. I showed you in the introduction that these immune cells are also present in the oral mucosa. They would need to reach these immune cells, so they would need to cross physiological barriers, but it is known that milk EVs or EVs can do this. So our question was, then can human milk EVs modulate cells? And we specifically looked at T cells. So the first part of the experiments that I will explain went like this. We isolated pure CD4 positive T cells from adult blood, and then we stimulate them with antibodies directed against CD3 and CD28. So this activates the T cells. And then we culture them in the presence of EVs or the other controls. And then our readout becomes proliferation as a measure of activation, or for instance, cytokine production. So what we did is we labeled these cells with cell trace violet. And what happens if the cells become activated because of the antibodies that we add, they start proliferating and the daughter cells get half of the dye. So you see a dilution of the signal. And actually with the milk EVs, they looked far more similar to the medium than to the positive control. And our procedural control, the EV depleted, still showed a lot of proliferation. So that is here, that's depicted over here. So it seems that these milk EVs affected the T cell proliferation and they actually inhibited the CD4 positive T cells. Then when we added different concentrations of EVs, we could see a dose-dependent effect, because if we added the largest volume possible, there was the most inhibition, and the least EVs we added, the least inhibition there was. We also got a question from a reviewer, said, can you even rescue the EV depleted procedural control by adding back EVs to this control? And that is actually what we show over here. So the more EV sample we add to the EV depleted control, the more suppression there is. So one of the things that you can look at when T cells are no longer proliferating is that maybe they retain a naive phenotype and they do not become mature or memory-like. And that is exactly what we did. So for this, you look at the expression of CD5RA, which is a marker for naive cells, or CD45RO, which is a marker for memory cells. And actually what we saw with the EVs is that there were more naive cells because they express more CD45RA, and with the EV depleted, there was more RO and less RO with the EVs. So this indicates that these milk EVs, they suppress the proliferation and they retain the cells in an EV phenotype. And then lastly, we checked the cytokine production in these cells. And basically any cytokine that we checked was lower in conditions where they were cultured in the presence of EVs, which to me makes sense because we're trying to turn on these cells by providing them or giving them the antibodies that turn on CD3 and CD28. But yeah, they are suppressed by the milk EVs. So they do not proliferate, they remain naive, and they don't produce any cytokines. But there was some thing here going on because a few years ago, there was a very important paper published in the Journal of Immunology by Suzanne Gabrielson's group that showed that vesicles from human milk have immunomodulatory properties, and they specifically study T cells. However, they say that they found regulatory T cells, which would maybe fit these data because regulatory T cells in vitro do not divide. So we put quite some effort in determining whether or not we also induced regulatory T cells, but in the end, we are convinced that we do not see any Tregs in our system. And why is that? Well, if we culture PBMCs, so peripheral blood mononuclear cells, together with these extracellular vesicles, we can look for Treg markers, so markers for regulatory T cells. So they should express CD4, and they should have a high expression of CD25 because they require this IL-2 receptor, because they require a lot of IL-2 to survive. Then what we did is we checked not only FOXP3 positive cells, but they should also have been negative for CD127. So this is the most thorough gating strategy that you can do on Tregs. And if we quantify this, and in the end, we did many different donors to check if it's really true, but we didn't find any differences between EV depleted or EVs. So in our case, we would say that these T cells remain inactivated, but they do not become regulatory T cells, because finally we did the most important experiment, and that is we performed a suppression assay. So what you do is, in this case, we took our T cells that were primed with either the antibodies in the presence of the EV or the EV depleted control. Then we took them out of culture and we placed them as primed cells together with antigen presenting cells and responder T cells. And the responder T cells, they were labeled with a fluorescent dye. So what you would see in the control condition is that these cells are proliferating, also with the EV depleted control, but also with the EVs. And if in these conditions we would have induced regulatory T cells, we had expected that the proliferation would have been lower, which was not the case. So for us, no regulatory T cells, but really naive T cells due to the milk EVs. Now, when we looked into our proteomics again, we saw a strong correlation with the PI3 kinase ACT pathway. There was a lot of potential activity there with many different hotspots. Cell cycles seemed to be inhibited, while mTOR, so the protein synthesis, seemed to be activated. And actually, this can be found in our supplementary. The mTOR is also involved in FOXP3 regulation. So it does fit our data that we don't find any Treg induction. So in the end, we said, well, what is going on with these milk EVs? Do they have a direct effect or do they have a lasting effect? So what we did is we did a primary stimulation of T cells together with EVs or EV depleted. And then we took these prime T cells and we re-stimulated them again in the presence of dendritic cells. Now, if the EVs would permanently shut down your T cells, they wouldn't proliferate after the re-stimulation. But actually what we saw is that most of the conditions, they were proliferating just as well as the EV depleted control. So there's a really direct suppression of T cells. So apparently in a vivo situation, the infant is exposed to the EVs, and then immune regulation of the T cells might happen. But as soon as the milk EVs are gone, these T cells can respond to signals again. So in conclusion, what we show is that with our in vitro system, we can look in the oral cavity. We see that there's an enhanced epithelial cell migration in the presence of milk EVs. We see that there's a very specific TLR regulation because only the endosomal TLRs that we tested were inhibited. And what we also see is that there's an inhibition of CD4 positive T cell proliferation and activation. We used our modeling of the proteomics to get an idea about what proteins can do in milk EVs. So they contain many different proteins and they're likely to form clusters. And then if you look at these signaling cascades, it's not that there's just one part of the signaling cascade that is potentially affected by the milk EVs, but it might happen at different levels throughout the cell. So this is what I wanted to show you. So I'm now happy to take on any questions. Thank you.
Thanks so much, Martijn. And we do indeed have quite a few questions that have come in the chat box. I'm sure we'll probably continue to get some questions. So let's go ahead and go to those questions. I'd just like to tell everybody who has put a question in there to please be alert. As I come to you, I will ask you to unmute yourself so that you can speak with Martijn and Marca. So let's start with Artem Zhivolozhny. You have a question about some of the other components that may be here, particulates. Go ahead, Artem.
Yeah, biofluids such as blood and milk contain terrific amount of lipoproteins. And some type of such lipoproteins, like, for example, HDA, has a very similar size and density, EV. And my question is how you remove lipoproteins from your sample, and or did you check the presence of lipoproteins after sample purification?
Yeah, thank you for your question. Yeah, we actually did not actively check if we find any lipoproteins in the milk, but yeah, I did do a literature search on this, because when people work, for instance, with plasma, these lipoprotein particles are a big issue. But in milk, not a lot of people have studied it. I have found really old publications that do show that there are lipoprotein particles in milk, but how many particles there are compared to the other particles in milk, that is unknown to me at the moment. So I don't know if they form just a minor part. And I am not completely sure if we can get rid of all the lipoprotein particles. What I can say is we have this density gradient together with the size exclusion chromatography. So we know that with the size exclusion chromatography, we should at least get rid of lipoprotein particles that are in different size. And perhaps with the density gradient, we can get rid of the lipoprotein particles that have a different density. So I think, if we would have used a precipitation method or a direct pelleting method, I think your argument would be very valid that we might have contamination. But at this point, I'm actually not even sure if there are that many particles in there. But I have to admit, we have not actively checked this.
Yeah, thanks, Artem, for that good question. I know that lipoprotein particles are probably the biggest problem in blood, but we also find them in some other biofluids that are filtrates of blood. So, for example, saliva, which I think saliva is very relevant here too, and then thinking about some of the work that's been done on lipoprotein particle EV interactions. I'm actually going to skip through some of the questions for the moment and go right to Rudiger Gross, because Rudiger has a question I think that is very closely related to this, and that is, in the oral compartment, what about other EVs that may be interacting with these milk EVs? And I would even ask, what about the lipoproteins if they're there in the saliva, which we know they are? Are they potentially?
I was last year wondering, first of all, how specific the effect is to milk EVs in general. So if any EV does it, or if milk EVs are especially potent at it, but more specifically, how the interplay would be with EVs that are in saliva, because obviously those EVs will come into contact when the infant is feeding, and there's infant own EVs in the saliva, and they come in contact with milk EVs. And maybe even further, and maybe not so relevant, but there might of course also be bacterial outer membrane vesicles, both in saliva and in milk. So do you have an idea if there's an interplay here?
Yeah, that's a very interesting question. So the first part is, the assays that we showed, and you say like, yeah, is this really milk EV specific? Of course, other vesicles can also modulate T cells, right? I mean, this is not unique to milk EVs, but we have to see it in the context of the infant growing up. And there it is a physiological phenomenon that we try to explain. So in that sense, I think it is good to know that milk EVs can do this. And maybe there is different T cell modulation among mothers. And maybe mothers with a different health status have a different T cell modulation. So I guess it is important to know that, but it's not that I'm claiming or that we are claiming that milk EVs are unique to other EV types. I think there is certainly an overlap. I find your question about, is there an interplay with other EVs, very interesting, because obviously there is saliva there. And maybe, yeah, maybe these EVs enhance each other, or maybe there is another form of regulation. And that also adds to your suggestion that there are these OMVs present in milk, which we believe might very well be, because if there is microbiota in milk, these microbiota is also producing vesicles. Now, the thing is, are these a lot of vesicles? It doesn't necessarily have to be, because they can still be very potent. Now we are isolating the complete prep, so we don't know which vesicle type we are looking at, but it would be interesting to more discriminate the different subsets and maybe to look at these OMVs to see what they do, because who knows what they do, because otherwise, why would milk have this microbiota? Obviously to support the child and to seed the child with the microbiota, but maybe it has additional function. Yeah.
Yeah, and maybe to add to that, I think this is really a very important discussion to have in the physiological context. Like, is there a possibility that the saliva EVs also bind to the milk EVs? It was also close, for example, to one of the reviewers' question about the LPS. For example, like, is it a contamination of the procedure that we are using in the lab, or is it LPS that is present in milk, or is it even LPS that is specifically bound to the milk EVs? And if you look to the proteome of the milk EVs, you see receptors for LPS. You even see that EVs have toll-like receptor 2 on their own surface. So it makes it awfully complicated, but biology is very complicated. So the interplay between lipoprotein particles and EVs is not a discussion only on contaminations, but it is a discussion on functional entities that can be formed and what is then their role in the biological system. And I think by just now really separating out these things, we need also to think on how we can get that in the big picture. So I really like these questions, but it's extremely complicated to really do the proper research on, but I think that's indeed the next level to get it into place.
Let's move now back to the top of the list, where Phil Askanese is asking about antigen-specific EVs. Phil, could you elaborate on your question? And I know Lilia Paniushkina also had some questions along this line.
Frank Renigal is part of the group, and he's helped us describe light chain on the surface of EVs. And I wonder if that's been part of the program. You've got a lot to do.
Yeah, so we have actually not actively looked for the light chain, and also not looked for if they are antigen specific. You know, they do bear MHC, they do bear MHC class 2, they bear all these immunoglobulins. So it is likely that they are antigen specific. It is maybe, yeah, when you know a little bit more about milk literature, where there are some ideas that even food components are introduced into the milk and they are co-expressed into the milk, that something is happening there. So it could very well be. We have not actively looked at this. I don't know, Marca, if you want to add something on this topic.
Yeah, definitely. It's indeed an important question also from Phil, to see whether there is that connection also from Lilia, saying like, is there a real connection between mother and child also in antigen specificity? And it can be either something like with the light chain, or also with specific receptors and MHC and antigen presentation. But you need to go so much into detail, and the milk EV population is as such already very heterogeneous, and maybe not only from the mother, but also indeed from bacteria and maybe from the diet, that I am not sure whether we can reach that resolution to do these analyses. And we should push further on the single EV-based analysis, and we can try to approach it with flow cytometry. But it is really a challenge to do that. So also next steps, but important to consider.
I urge you to consider anti-light chain column affinity chromatography to separate out such a subpopulation. But you're right, the complexity is enormous.
Yeah, and if I can add to that, and already sort of anticipating further discussion, I see that Suzanne Gabrielson also had a question about this. Maybe you can immunocapture specific subsets of EVs to further analyze them. And I think that is interesting approach to look at these subsets, or in response to Suzanne's question, to really deplete EVs from preparations. So I think, you know, for this study, we wanted to first look at the bulk of the EVs to see what they do. Now we have these in vitro culture systems in place, so now we can play around with that. So we greatly value your input in this, because looking into subsets, I think, is the next step into this milk EV research.
Maybe I can also, once you're addressing me, also just comment on this comparison with the Tregs. I think you have a point there. What we saw was inhibition of IL-2 production from T cells, and we tried to explain that. But back in 2007, what you did was that you only looked at FOXP3 and CD3, CD4. So it was a simplified way, and since then we know more how to really determine Tregs better. And also, as you did the functional assay, that is also a better proof that you don't really see upregulation of T regulatory cells.
Yeah, and I think that the thing that you had with your publication is you were the first one also to include proteomics, also to include functionality. So that was already such an important paper. We tried to move on, and there are still many questions that we have not answered in this paper, but you know, the paper is not an endpoint. You know, we need to do more research, and we've had this question numerous times, obviously. Why don't you see regulatory T cells while another group has seen it? The reviewers asked for this as well, but we already have very different isolation procedures, which makes it difficult to compare. Then you could go into the biology. So that's why we stimulated the PBMCs and not our pure T cells, as we were used to doing, because we wanted to have sort of a cleaner system. But there are so many differences there that it's really hard to compare. And I think that that is something that the entire EV field is obviously struggling with. We have all these different isolation procedures and then these different readouts. So in the end, we'll keep on having these discussions, but I think it is always good to move forward. So maybe you did.
But I also think, I mean, there is also a point that you do have so many other stuff in the milk. So if there are other contaminating factors that induce Tregs, and you have the exosomes carrying, for example, food antigens, the net effect might still be that you also have Treg induction. So
and in the end, we were also struggling with the amount of milk, obviously, that we have to perform experiments with. So with this procedure that we have now, we know for sure we have a procedural control for the OptiPrep and the SEC, and it is sort of in a protein background. But otherwise, we have to use twice the amount of milk sample to get your EV, and then your EV sample, as you would approach it, you will need to deplete it, which is also dependent on how efficient is the binding of the EVs to these beads. So in the end, we also make a pragmatic decision to say, okay, this is the amount of milk that we have, and we cannot use milliliters of milk because it's hard to come by. So in the end, these controls are difficult. I'm happy that we at least included this control already, and we're continuously thinking about how to improve this. And your suggestion is well taken. Thank you very much.
And
I wanted to encourage them to look at effects on antigen presenting cells in the looking they're doing at the immune system.
It's ongoing, Phil.
Good.
Great. All right. Well, I think there's also a question, and in the interest of time, I'm just going to cover a few of these questions myself. So Jagdeep Sandhu asks if you checked for FOXP3 negative Treg population.
Yeah, we also, obviously, when you do the stainings, you have all the stainings there, and there were no major differences there. So yeah, that's my short answer to that question.
And then question about the flow cytometry. So can you just briefly mention the flow cytometry that you used?
Yeah, this is just a regular flow cytometer, right? Just to measure the cells. So this was a Canto by BD, and nothing fancy. You just check the cells and you do your staining. So that is hopefully all very well explained in the material methods of the paper.
Great. And a question about the proteomics. So Rob Hollenbeck asks, why did other proteomic studies in milk miss some of the proteins that you found? And is this because you have enriched for the EVs? Is it because EVs were lost from previous studies?
Yeah, I think it's the enrichment, because if you have a few very abundant milk proteins, which you have, and if you would measure a whole milk sample, the detector constantly measures these abundant milk proteins. So the less abundant milk proteins are masked by these few abundant proteins. So we just have a very good separation procedure, which allowed us to identify so many new proteins. And also we were, after Suzanne Gabrielson's group, the only ones to do it on human milk. So our resolution, or yeah, there was not much data already known. Together with our high resolution, gave us this 633 unique proteins that no one had identified before.
All right. Harry, do you want to ask a question about species differences?
Well, thanks, Martin. This was a brilliant presentation. Thanks for a newcomer to the milk field. Really, my question is whether you have had the opportunity to compare the human and cow milk, and how could we possibly get similarities or different signals from those?
Yeah, that's a very intriguing question, and I would refer to a paper in the Journal of Nutrition that we also published this month, because there we look for in bovine milk and we compare raw milk to industrial processed milk, and we saw that pasteurization does something to EVs. This is not yet functionality, but it is on just basic characteristics. But it could be that because we ingest not raw milk, but pasteurized milk, this does something different than if you would ingest raw milk. And I also have an allergy background, which also this fascinates me, because it is known that children who consume raw cow's milk when they live on the farm, they have less allergies than children that live on a farm, but they consume pasteurized milk. So I think that there is a level of regulation by the EVs. I have not proven this, but I think that there might be something going on here. What we do see is that when we take our human T cells and we perform the same assay with bovine milk EVs, they do the same thing. So there is this same regulation, and that could be because of their content. We have also compared in one study that we published the microRNA content from different species. And there we show that there are a couple of microRNAs that have identical sequences that are found throughout the different species.
Wow. Wow. This is fantastic. Really nice. Thank you so much.
Thank you very much.
Thanks, Harry. And so Ali Reza Fazeli asks a question that is kind of wait a second, what does this mean? So why would you want to dampen immune responses? Does it have to do with developing allergies? I mean, what is it that you think is the reason for this?
I think this is a homework assignment for all of us. What does this mean? I mean, it can mean so many different things, and I don't have the answer to it, but it's such a specific regulation. Why do you regulate endosomal TLRs but not cell surface TLRs? Why do you regulate these T cells, but at the same time you enhance barrier function? And it is a natural phenomenon. So we do these in vitro assays. So let's assume that it applies to the in vivo situation. Sometimes maybe immune activation all the time is not beneficial. Maybe you need some regulation. And as we've shown with the T cells, I think the restimulation assay is really nice because it shows with the direct presence of the milk EVs, there's regulation. If they are gone, the cells are responsive. And I think maybe also for allergy induction, it can be of importance. Maybe, you know, when these children are drinking milk and it's mature milk, they're getting their first bites to eat and they're sampling their environment. And maybe you do not need to react to environmental signals. So that's why maybe the milk EVs are providing this signal, but I'm not completely sure. It's so complicated and it's sort of counterintuitive. I get the question, but yeah, I think immune regulation apparently is also important for the newborn.
Indeed.
Yeah, maybe to add on that, I think immune regulation is one of the most crucial things we have in our body for all the diseases that we encounter also in later life, infectious diseases, cancer, etc., etc. So if we do not have a good immune regulation, we cannot survive properly. And that is for all mammals, it is the case. So our hypothesis is really that when the newborn is there in the new environment, it is exposed to all these different triggers. The whole microbiome needs to be formed. You are exposed to all other triggers around there. So our hypothesis is that the milk EVs present a temporal window in which the system can adapt. You do not want to have an immune system going crazy once the child is born and is exposed to all the new things. Then you have a real pro-inflammatory profile and you get cytokine storms, etc., etc., like also people now with COVID are really having. So we think that these milk EVs really are doing the balancing act and that they play a role in the building of a resilient immune system. So you need a bit of CXCL8, for example, to develop the gastrointestinal tract. But if you have a lot of CXCL8, then you have a big problem there. So it is really learning to balance. And that is what model provides to the child, how to learn to balance.
But it is a very, very specific reduction. You are reducing just the TLRs, that endosomal, and mainly viral. Can I just ask one question, that how short was the time, that shortest time that you were able to see this effect? I mean, how long does it take? You treat your cells with the EVs and you would see this effect.
So for the TLRs, this was 4 hours. For the epithelial cells, for the migration, it was 3 hours. And our T cell assays we performed for 6 days. And that has to do with how long does it take to see the difference in proliferation and also in cytokine production.
But for the messenger RNAs in the T cells, we also look after a couple of hours and you already see the different profiles. So I think this is an important question. Like what we see is that the milk EVs have an instantaneous effect. So it looks like it is less doing on transcriptional regulation, translational regulation. So that was the reason why we immediately also looked to the proteome. We said, if we add the EVs together with the stimulus, we directly see an effect. So it should be something that kicks in immediately. And for us, it was the most logical thing then to first analyze the proteome because you have these things available. And I think that is really something that was very peculiar. We did not pre-treat the cells with the milk EVs. It was just, we give it at the same time point and you see the effect, very strong. If you remove the milk EVs, then it's gone. So it is something like a direct effect. And I'm really puzzled with it, but that is how the action looked.
Very good. So we're at the end of our hour, actually a little bit over, but I do want to quickly go to the questions from David Armstrong, who asks, you told us that you are normalizing basically by the amount of milk that you start with, but do you have an idea of what the EV to cell ratio might be in your treatments?
Yeah, so I don't know this by heart actually, but we know from the particle numbers that we got from figure 1a that it's about, I think, 8 to the 10 to the 12 particles per ml. In a T cell assay, we add 750 microliters, so we could calculate back, because we we culture 500,000 cells in a well, how many EVs that would be per cell. So we can make a rough estimate about what that is. I have to admit that I don't know how that relates to other research, if other people use more EVs or less EVs. So I'm not sure about that.
Good. Okay. Well, we'll have to end it here. We have some additional questions. You know, what are the components that are causing these effects? What might be the different effects of EVs at different stages of lactation? So I think that a lot of good questions have been raised here.
Fantastic.
And it's wonderful to see the progress that you've made, and to think about all the different places that we could be going in the near future with studies of milk EVs. So Martijn, thank you so much for your very clear presentation and your answers. Marca, thank you so much for supervising this research and really driving the field of milk EVs forward. And thank you all for joining today. So I'll just conclude by reminding you that the ICEF 2021 meeting is coming up in May. There's still time to register for it if you haven't. So I hope to see all of you at an upcoming EV club or at the ICEF 2021 meeting. So thanks again, everybody, and take care. Bye now.
Thank you.